Yani chairing the fifth session for today on artificial intelligence applications in medicine. Chairing with me will be Prof. Hani Timsah, inshallah. For our first speaker, we have Dr. Khulud Al-Yahya. She holds a PhD in computer science from the University of Birmingham and currently serves as the director of the Digital Innovation Unit at the Entrepreneurship Institute in King Saud University. Her research focuses on developing innovative AI methodologies to tackle complex data-driven problems. She contributed to numerous journals. Alongside her academic work, she has led data-driven projects that bridge the gap between theoretical research and practical applications. Dr. Khulud, the mic is yours. You can go ahead and proceed with your lecture for today.
Thank you, Dr. Noura. Thank you for the kind introduction and for having me here and allowing me to share my research. So I'll share my screen. Can you see it as a full screen?
Not as a full screen.
Okay. How about now?
It's the same.
Okay. Is it still the same?
Yes.
Okay. Sorry. To be honest, Canva, this is my first time using Canva. I usually use Oh, okay. Someone is saying that they can see it as a full screen. But let me see an alternative. Now, can you see it as a full screen?
It's not the full screen, but it's decent. I mean, you can proceed. Honestly, it's fine.
Okay. Okay. Hello, everyone. Today I'll talk to you about an application I'm developing with my student, their final year students, and IT, information technology department. We're developing an application called Insulin Sync. The aim is to have a mobile application powered by AI to help diabetic patients manage their diabetes. So this work is with my brilliant student, Warif Youssef, Reem Hawass, Ranim Al Yahya, and Hind Al Hijailan. So I'll start by talking about like the current status about diabetes and why there's a need for such application. So the World Health Organization have reported there is a drastic increase in diabetes prevalence across all income levels. Now it's around 9% of the adult population have diabetes. It's expected that this number will raise to 630, around 30 million by 2040. In 2015, 1.6 million deaths were attributed to diabetes. By 2030, it's projected that diabetes will be the 7th leading cause of death. In Saudi Arabia, I think the matter is worse, and it's almost a national crisis. There's some research that label diabetes as epidemic in Saudi Arabia and projected that by 2030, around 50% of the population, of the Saudi population will have diabetes. So the current challenges is there's a lack of urgency in reversing this trend. And there is, well, which is the focus of this application, there is a growing complication associated with the health risk due to poor management of diabetes. And this poor management, I get to experience firsthand, and it was the reason why I had this idea for the application. One of my family members, adult family members, got a late onset of type 1 diabetes. And trying to just manage it was really overwhelming. And that's no surprise. Some research shows that there are 42 factors that can affect your blood glucose level. So for a diabetic patient, type 1 diabetic patients especially, who are reliant on insulin, and especially the ones who take multiple daily injections, they have to take multiple decisions during the day about their insulin dosage, their current glucose level, what affected that current glucose level, and they have to get it right to avoid complications such as like hyperglycemia or hypoglycemia. We interviewed 151 type 1 patients aged 18 to 65. 55% of them said calculating insulin dosage is challenging. It's even more than that if they were recently got the disease, like within 1 to 2 years, they still didn't develop this intuition of what's the right insulin dosage. 65%, a lot. So the insulin dosage, there are many factors. One of the factors is the meal you eat. And the current standard is you only count the carbohydrate in the meal to estimate the insulin dosage. 65% of them said that calculating the amount of carbs is challenging and tedious sometimes. Only 21% of the people we interviewed said they get the insulin dosage correctly. So there is a need to help them calculate the appropriate insulin dosage to avoid misjudgment. The current methods, there are some current methods, there are some advanced methods like insulin pump that have an algorithm in it, artificial pancreas, some of it is hyper closed loop. Unfortunately, not everyone has access to that. So a lot of patients still rely on multiple daily injections. So still they need help in making this decision. The other issue is most of the existing method, even the pump, all these advanced closed loop system, when they are calculating the insulin post meal, or the insulin related to the meal, they usually only account for carbohydrates. But there is research that shows actually fat and protein impact blood glucose significantly. So there is a need for a tool that considers all micronutrients in insulin dosage. So you can imagine that counting carbohydrate is already difficult. How about counting all the micronutrients in a meal? So here is where we think AI will be a great tool to help. So our solution, these are screenshots from our current application. So our solution is having an AI model for insulin dosage recommendation. This is being developed during this semester of the project. So AI is very good at analyzing large amount of data, picking patterns that we can't see, and this is available. Like they have the app, they have continuous glucose monitor, or even if they don't have a continuous glucose monitor, they usually get this data daily. So why not just use this data, use the trends with their meals, with their insulin, with their exercise, to build a better recommendation system, personalized recommendation system, instead of what they usually get now when they go to their doctors. Their doctors usually, they give them some suggestion for the insulin dosage that they should take. And that suggestion usually starts with a baseline that is average across all patients. But now with AI, we have the power to personalize it. So that's one of the components on the application. We have an AI model that is learning from the patient data and give them personalized recommendation for their insulins. This application integrates with the sensor. If they have a continuous glucose monitor sensor, they integrate with it. If they have a fitness tracker like Apple Watch or Garmin Watch and they do exercise, we take that into account in the calculation of the insulin. We also have, we said like we have identified that counting the micronutrient in a meal is very, very challenging and very tedious. So we offer them a number of options a number of easy options to count these macronutrients. We give them an ability to, if the meal has a barcode, to scan a barcode, then we consult a database and give them the micronutrients. Or we can give them a database. We give them a database. They can search the meal. They get the micronutrients. But the most advanced AI model that we were working on now is a computer vision AI model that you take a picture of a meal and then it counts the mass of the meal and it counts all the macronutrients, protein, fat, carb, and so on. So they have a number of options to count the macronutrients in a meal, and hopefully that feeds in our AI recommendation system for insulin dosage and will help them just count it, like it help them with behavior. There are behavior issues with managing insulin and logging that, and one like making this easier will help hopefully with this behavior issue. We have daily logs showing everything, all the data they have entered. We have alerts and notification as part of the system. So I'll not take too long. We compared with existing apps and we found a gap. So what we are offering is a mix between what some diet application offer and some diabetic application offer. So we offer all of the features. I'll not bore you with the architecture design. Currently, we are only working with Freestyle Libre because almost 70% of the patients said they could use Freestyle Libre. It's because it's supported by the government, it's cheaper, it takes less time between readings. So our users, the user we have tested the app with, are type 1 diabetics with multiple insulin daily injection. They're 18 years old and older. They should just have a basic understanding of English, some familiarity with using web application, and use Freestyle Libre to be able to use our app. So we tested our app, the first few features that we implemented in the first semester. And the demographics of the diabetic patients that we tested with, they were between 31 to 45 years old. They all had bachelor degree. Their technical expertise, they said it's medium. And it's important that we address we design an app to be accessible to all technical expertise level. They said they were familiar, 100% of them said they're familiar with similar apps. So we gave them a bunch of tasks to complete in the app, and all of the tasks were completed. On a usability scale test, system usability scale test that we usually use if we have a system and we want to see how easy it is to be used, we got 92%, and this is really great because the minimum for such test is 68%. And most importantly, 96% of the users, they said they would use this app frequently. And that's what we want to do. And we're trying to implement our feature such that it's easier, makes people able to use it. We had a few issues, but we fixed them. In conclusion, Insulin Sync, this app aims to revolutionize how the type 1 diabetic management is managed through AI and offering personalized care. We are empowering users by making simplified calculation of optimal insulin dosage. So every dosage we give the reasoning why we reach this recommendation. And to be honest, this was one of the trickiest things, because with AI, things can be complicated and can be a black box, and we don't know how they reach this recommendation. This usually is frowned upon in medical applications, because we want transparency in case some of the calculation is wrong. So that's what we are trying to achieve. So we are providing real-time guidance. Our aim is to reduce the reliance on manual calculation and to support individualized treatment. So with this application we are currently working on 2 research papers. One is for the computer vision, the advanced model for computer vision that quantifies micronutrients. The other one is an optimization algorithm that takes into account most of the factors that we can collect about the patient to give a personalized insulin recommendations. We need help further later on once we finish the application. We're now testing it in a data of a single patient and artificial data and simulation. So this is not enough. We need to do, after we finish the application, we need to do a randomized control trial with type 1 diabetics with multiple daily injection therapy to see if it actually improves their blood glucose management. So if you're interested, please get in touch with me on this email. We're also in our model that quantifies micronutrients from images. What we discovered, what we're facing, is there's a lack of data in Saudi cuisine. So we are currently collecting data about Saudi cuisine. So if you want to help my student and help us collect more data and be mentioned and acknowledged, please scan this QR code. It will take you to a Google form where you can upload. Even a single dish will be helpful. Also here, I'm calling any dietitian. If you want to collaborate in this project, please get in touch with me. There's a room for collaboration with dietitian. Thank you for your listening, and sorry for the technical issue.
Thank you, Dr. Khulud. There were no technical issues. Apparently, I had an issue in my Zoom display. This was very, very informative at both a scientific and a personal level. Thank you so much. We'll proceed with our next speaker, Dr. Rayyan Al-Fallaj. He is a 5th-year postgrad resident in otolaryngology head and neck surgery at King Saud University Medical City. His primary interests in research includes the application of artificial intelligence within the field of rhinology. He has authored several publications and remains actively engaged in advancing knowledge and practices in the otolaryngology specialty. Dr. Rayyan, the mic is all yours. Dr. Rayyan, we see your screen, but I can't hear your voice.
Assalamu alaikum. Sorry.
وعليكم السلام ورحمة الله.
Thank you, Dr. Anora, for this introduction. I would like to thank Professor for the kind invitation. My name is Rayyan Al-Fallaj. I'm a postgraduate 5th-year resident at laryngology program in King's University. And it is truly both an honor and privilege to be here to present our project, um, machine learning use of prediction models in a CSF leak, cerebrospinal fluid leak, and skull-base reconstruction. And as we delve into the forefront of research and advancement in artificial intelligence, this project, at least in our field, represent a remarkable leap forward in applications of artificial intelligence. And I would like to take a moment to acknowledge my research team led by Dr. Ali Zershade. He's the chairman in our department and assistant professor in otolaryngology. And we had a collaborations with College of Computer Science with Prof. Yaqub Bilhazi and Prof. Muhammad Al-Rahal. And their significant collaborations with our project were a key to success and to do this project as this marks a pivotal milestone in our journey to elevate the interdisciplinary research and innovation. So I have nothing to disclose, and I want to just brief introductions about the topic. We have Indonesian approaches in skull-based surgeries where we do the our procedures in collaborations with the neurosurgery in cases where we remove tumors from the brain through the endoscope through the nose. Where they do the procedures first, we open the approach, the corridor from the nose into the skull base from the sinuses. After removing the tumor, we will be having a defect into the skull base that we need to reconstruct because opening that defect and removing that tumor will be uh, there will be a CSF leak, cerebrospinal fluid leak from the meninges where it is the boundaries of the brain. These this situations where we have a leak after the reconstructions of the defect is very crucial and can have very high morbidity and mortality due to the risk of meningitis and death. So, what's our area and our role in that surgery? We do the reconstruction of the CSF leak. We use multiple materials and multiple approaches to reconstruct that defect. However, there are a lot of factors can contribute to the success or failure of that reconstruction. So, what is the role of machine learning in that aspect? Is to predict. We want machine learning to predict our factors that lead to success or failure. Machine learning and artificial intelligence processes vast amount of surgical data, which were shown in the literature, and it shown its ability in prediction models, and that can give us the results based on multiple factors, and can have also enhanced the preparations of the skull-based reconstructions through integrations and planning of the procedure. So we did a literature review for that aspect, and we found multiple studies. One of its largest of its kinds done in UK, where they did neural network and they compared the neural network to logistic regression and decision tree in prediction of a CSF leak post Indonesia skull-based surgeries. And neural network outperformed the logistic regressions and decision tree for the CSF leak predictions with 88% accuracy, and they had 83% sensitivity and 89% specificity. Another studies, they use it in pituitary adenoma surgeries in predictions specifically. Surprisingly, the traditional statistical methods found no risk factors for CSF leak were identified. However, in using a neural network, they found that 89% accuracy and 83% sensitivity. We want to do something different because neural network is a very sophisticated approach that needed pre-processing of the data. We had an idea that we want to use our data, which is ready, and we collected the data. However, it is in qualitative form. We want something to deal with our data as it is. We don't need to convert our data to numerical data. So we developed a language model based for CSF leak prediction. And we want to use the natural language processing and straightforward dealing with data to take our data and give it the predictions and the result based on that as it is without converting the data. So our study was done in King Saud University Medical City. It was a cross-sectional validations study, and we included all the patients who underwent nasal skull-based reconstructive surgeries after removal of the tumor from May 2015 to July 2020. We tried to include all the study samples we had in that period in order to maximize the sample size and empower the study. Um, this is just a quick look into the data we had. It is a lot of data and it is very variable data. We had an information about the patient's demographics, age, gender, BMI. We have perioperative suspicions, clinical suspicions of a CSF leak based on the symptoms the patient having, based on the length of stay in the hospital, the timing of the CT scan that it is done after the procedure to double check if there is any signs that can tell us that there is a CSF leak or not. Pathology-related factors. What was the tumor? Is there any hydrocephalus? What's the tumor location? The histopathological grading of the tumor. What is the surgical factor, intraoperative factor that we did inside the surgery? What was the approach? Is it extended approach or limited approach? Did we use a lumbar drain, which is the CSF diversion method? Was there any intraoperative suspicion of CSF leak or findings such as CSF leak flow? Oh, and what material we used in the reconstructions? If we look into the literature, there are a lot of things and materials we use, such as nasal backing, fat, fascia, muscles, a lot of things with different success percentage. So, and also we have perioperative CT scan findings, informations about after the surgery, such as the timing when we do the CT, is there any features in the CT scan that tells us that there is a leak or not, such as air in the brain or hematoma or a defect. So these data are a lot of data, and it's very difficult to predict which one is contributing most to the CSF leak. So first of all, we brought our biostatistician and we gave him the data, and he prepared a model for logistic regressions in order to compare the natural language processing with logistic regressions. And he developed a logistic regression model, and he calculated the data and percentages and the predictions of CSF leak based on logistic regression. And we developed the language model. We're using the BERT model. BERT model, it's dealing with the categorical medical data as it is, and this side is like as we look from inside the BERT model, which is automatically converting the tubular data sets and tokenizations, giving tokenizations of that tubular data set as it is converting the qualitative data into numbers inside that model. We don't need to convert the qualitative data into numbers. It is automatically converting the data. And it has a feature on top of that, a classifier to classify the result, is it CSF leak or not. Plus, on top of that, it has attention weight of each variable. It has a feature that gives us the attention weight, which is the most important for us in practice. We want to know if that patient carry the most significant risk factor for CSF leak or not. So, for example, the patient obese with a BMI more than 48, we need to have a different plan for that patient because he's having high risk of CSF leak. So this is the architecture, what we had. We gave our biostatistician the data to compare with the language model, and all the factors were collected, and we split the data for the BERT model into training 50% and 50% for testing. So this is just descriptive data that we had, and it was 115 patients, 16 patients, and 15 patients who had a CSF leak, and 101 patients who didn't have a CSF leak. So what about the performance we found with the BERT model in compared to logistic regressions? The accuracy, BERT model had a 96.67% of accuracy compared to 72% in logistic regression. It was 98% sensitive BERT model. However, logistic regression was 21%. Specificity were specific in both models, and F1 score in BERT model was 90%. So as we see here, the BERT model outperformed the function of traditional logistic regression. What's the key finding we found in both models? Sometimes also in the literature, they mentioned that traditional statistical method can find factors that were not found in the artificial intelligence model and vice versa. So logistic regressions, they found that preoperative hydrocephalus had 5 times higher risk for CSF leak, fat reconstruction associated with 20 times higher risk of CSF leak, and overweight patients have 7.15 times higher risk of CSF leak. However, in BERT model told us that CT scan is very crucial for that patients. Postoperative CT scan features most predictive of CSF leak. If there is a findings in the CT scan, it is the most predictive. And the surgical factors were the second most important group in models decision making. And it is the factors that decide the success or the failure of the reconstructions. And the model can consider multiple factors for accurate predictions as we saw. So, and this is the attention weight that it gives us in the BERT model, as we see the perioperative CT scan and surgical factor. So the key findings we found in BERT model that AI has a superiority over the traditional statistical method. It outperformed the conventional statistical method, and it showed us its abilities and great abilities in handling qualitative data and large amount of diverse data with multiple factors, and it can also predict the future. CT scan features and surgical factors are important predictors for CSF leak, and the BERT model has shown its abilities in prediction. So, as a conclusion, AI integrations and machine learning enhances the predictions of the surgical outcomes, and its integrations in our practice is very important in changing our practice and enhancing the success of our practice. The model outperformed the traditional methods in identifying CSF leak, and as we mentioned, CT scan and surgical factors were crucial for predictions. And these are my references, and thank you. I'm happy to receive questions.
Thank you so much, Dr. Rayan. This was actually very, very impressive work, very informative. Appreciate it. And you finished ahead of time, which gives us some leeway probably for the next speaker. Thank you so much. Inshallah, the audience will be connecting with you in the networking session. So if I may proceed to introduce our next speaker, Prof. Hasib Khan is a distinguished professor at the Department of Biochemistry, College of Science at King Saud University. He is a group leader of Analytical and Molecular Bioscience Research Group and chair professor at Research Chair for Biomedical Applications of Nanomaterials. He has made significant contributions in bridging gaps between chemistry, biology, computation, and medical sciences. He published numerous publications and developed software tools for biomedical applications, and he is listed in the top 2% world ranking of scientists. So, Prof. Hasib Khan, if you are ready and available to go ahead and proceed with your presentation. Just a note for the audience that Prof. Hasib Khan will not be able to join us in the networking sessions. So if you have any questions, please feel free to write it in the Q&A section, and hopefully we will have time to go through them at the end of presentation. Prof. Hasib, you can go ahead.
Thank you very much, Dr. Noura, for introducing me to the attendees. My sincere gratitude and appreciation to Professor Asma and her team for organizing this consecutive fourth symposium of CMMM. Actually, I'm a biochemist, but I have interest in computer programming. So today I'm going to highlight the innovation, what we have done to resolve some issues when we calculate this Fisher exact test probability. I think you may be familiar with the Fisher exact test. It's a century-old test. And that time, even there were no calculators and computers and people. They were using this like paperwork only for calculating the probability. And there is another test which is called chi-square test. So in this presentation, I'm going to compare both the tests, how they are calculated, when they should be used, and what is the innovation that we have made to resolve the computation of this Fisher exact test. This is mainly both the tests, Fisher exact test and chi-square test. They are used to calculate the probability relationship between two dichotomous variables. I have shown here the setting of Fisher exact test. The same setting applies to chi-square test as well. So there are two rows and two columns. Here I mentioned the two exposures can be two different groups, and the outcome can be yes, no, can be live, dead, or can be the cancer relapsed, not relapsed. So any two dichotomous variables will be there. When we see the computation method, it is given here. If you see the equation, p-value is equal to t, and there is factorial sign. So this is fairly straightforward formula, but it is very tedious when the sample size is larger. If we see in the left side, so these are the values, x1, x2, x3, x4. So these are the frequencies. And the t is the total of rows. And here, if you see T3 and T4, these are the total of columns. These values are put in the formula, and we get the p-value directly. This is the distribution-free test. So there's no table to be used for calculating Fisher's exact probability. However, there's a table in chi-square test. So there are two problems here. If the sample size is very large, it will be almost impossible to calculate p-value using handheld calculator or even computer. The second problem arises when the frequency, the minimum value is not zero. If it is more than that, so we will have to use extra tables to construct more tables. For example, if the minimum value is 10, so we will have to construct extra 10 values. So the original we will calculate the original p-value plus additional 10 more p-values, and the sum of that will be the corrected p-value. So it is very tedious. However, if we see the formula of chi-square test here given in the bottom, it is very simple. So there's no factorial computation. Just, I think there is only subtraction sign, and this is the division sign. So any number of values can easily be computed using chi-square test. Both these tests, they do the same job. They are used for the same purpose, but the calculation principle is quite different. It's very different. So chi-square test is an approximate test. It cannot be considered as exact test. This is the main difference. Whereas Fisher exact test, it gives exact probability value, and chi-square test gives approximate p-value. However, there are certain conditions. For example, this chi-square test must be avoided if there are few observations. If the observations or the sample size is less than 10, so it should not be used. Otherwise, we will get the incorrect p-values. However, Fisher, this is FET is short for Fisher exact test. It should be used, preferably should be used always. However, it must be used when the sample size is small. We can see the problem here for computation of Fisher probability. The conventional formula of Fisher exact test can only be used for a total of 113 subjects. If we use a computer, we cannot use this formula for more than 113 subjects. The one thing is that you will not see any factorial sign. I think factorial sign is there in the keyboard, but we cannot apply directly. Even if we use in Excel, it will not work. Even if we use in the calculator given in the computer, in accessories, it doesn't work. It's only the symbol, factorial symbol. When this I tried using handheld calculator, so it works up to 69. So the factorial 69 is 1.7. And these you see are 98 zeros. That means times 10 to the power 98. Beyond that, it doesn't work and it gives error. I tried this factorial 70. So we cannot calculate factorial 70 using handheld calculator. So this is the limitation. So what we did, we developed equation, and this equation is based on logarithm. Here you can see this is the log-based equation. So how to compute the p-value? All the values are converted into logarithm, starting from the minimum frequency, and this is from 0 goes to minimum. So we have to construct these number of tables starting from the minimum frequency up to 0 when the cell value becomes 0, and then to sum all the log values, and then finally take the anti-log of that. So this is here exp. This is the anti-log. Here, when we did the preliminary work, so we found that the upper limit of Visual Basic, this is the programming language, it is 1.79 times 10 to the power 308 zeros. This is, if we compare with the calculator, it is 3 times. This is the capacity is 3 times larger. And it is roughly close to factorial 170. That means if we use a computer, so we can do the factorial of up to 170. Beyond that, it will cross the computer limit and the computer will hang. Then we developed one software based on that equation. The software itself, if you see, the size is just 48 kilobytes, very small size software. However, it handles, you see, a lot of zeros, means more than 300 zeros, it can handle. And it will work on any Windows environment, even without the presence of Visual Basic program. It's not necessary that the Visual Basic program must be there on the computer. It will do any Windows, starting from Windows 7, 8, 9, 10, whatever. I will try to show if it can work here. It looks like, just cover this. So simply we have to enter some values. I will enter more than 100 to show. And this so simply it gives the p-values, both one tail and two tail. Actually, calculating two tail value is again very difficult task. it requires an additional tables in the opposite direction. So the computer has to do double work. Here, we can reset this. This is the history. We can show the history or clear the history. And there's a help topic also here. So this help is in Russian language, and this is in English language. And now I'm going to, we here, I tried up to 10,000 frequencies. Then I put
Prof. Hasib, I'm not sure if it's just me, but I'm seeing blank. I don't think I can.
yeah, this I was expecting the same actually.
Yeah.
But here I can see yes,
Okay.
it is possible, yes. So that means you didn't see the software functioning.
No, I mean, I just
yeah.
see the shadow of
Yes,
your
yes,
window or the
yes,
rectangles, but I don't see the
yes.
calculations,
I think
unfortunately.
it's not possible in Zoom. I'm simply exiting this.
And I think, are we on time still?
So
Yes. His time he should have until 11:20.
then we validated the software using our software compared to SPSS, and there's another software that can do Fisher's exact test, that is EPI info free. This is freeware, freely available software. So from here we can see if the values are few, the total number of subjects are less, then both these software they can perform Fisher's test, and our values tally both these software. However, when the frequencies are larger, so they cannot do both these software, they cannot perform Fisher's test. This NP is not possible. Whereas Fisher program, it computes the p-values. I mentioned here SPSS calculates Fisher's exact p-value only when the subjects are 20 or less. So even if we give the command in SPSS for Fisher's exact test and the sample size is larger, automatically it turns to chi-square test. It will give chi-square values. Whereas this software API performs Fisher's test when any of the expected values less than 5. This is the condition for API software. So this is about the internationally. Actually, we received a lot of requests from different institutions. From Tom's State University, they offered free hosting of this software. They translated also the help topic and they hosted there. And the formula was also used by the scientists from Emory University, USA. We received the request from them. And then they created also in their website also, they mentioned that we got this formula from us. Again, the scientists from Brain Science Institute, so in University, Australia, they also used our formula. And the scientists from National University of Singapore, they also developed one software based on our formula. And this software is dealing with microRNA pathway analysis too. We also received some feedbacks from the scientists who used our software. This from Emory University, from Schoenberg University, the National University of Singapore and University of Queensland. These were the students, and they developed their own software based on our formula. This is a few more. And, thank you very much for your patience. If you have any questions, please go ahead.
Thank you so much, Prof. Hasib, for this presentation. For now, I don't see any questions in the Q&A section. Does anyone from our panelists have any questions for Prof. Hasib? Okay, then thank you, Prof. Hasib, for your time. I would like to ask Prof. Hani to continue chairing the remaining of the session, please.
Okay, thank you very much. I think now we will move to our, just to ensure there is any questions for Prof. Hasib or none. I cannot see, no raised hands, no, or no, nothing in the chat so far. Okay. So we will be moving now to our next panel discussion about the role of machine learning in detecting small and large molecules toxicity in drug development and regulatory affairs. And it's with our colleagues, Dr. Ahad Abu Shal, Dr. Faiza bin Jubair, and our moderator will be Prof. Sarah Rushud.
Thank you, Prof. Hani, for the introduction. Actually, I thank Prof. Hasib for his informative application, computational dynamics and details that we work together. And I would love to introduce my colleagues, Dr. Faiza bin Jubair. She is an assistant professor in pharmaceutical chemistry department at King Saud University, and she has her PhD in pharmaceutical chemistry from School of Pharmacy and Pharmaceutical Science at Cardiff University at UK. She's specializing in drug docking and designing and synthesis of the novel CIP inhibitors. Her research work focused in computational studies to understand the ligand-protein interaction and improve its affinity, activity, or resistance using different techniques. Furthermore, Dr. Faiza had experience developing a homology model using a combination of homology modeling, molecular dynamics, simulations, and molecular docking to understand the active site and the binding interaction of selected ligand complexes. Dr. Faiza is also interested in drug designing, discovery, drug metabolism studies, and development of novel molecules, medicine inhibitors, and multiple targets such as antimicrobial agents, anti-diabetic agents, anti-cancer drugs, and pro-drugs design. And my other colleagues is Dr. Ahad Abu Shal. Dr. Ahad holding a Master of Science degree in pharmaceutical biotechnologies, and she is a senior scientific evaluation expert at Saudi FDA. And where Ahad, she's responsible for the evaluation of the quality of the active pharmaceutical ingredients, APIs, developing the SOPs as guidelines and quality inquiries, and improving the drug evaluation process. Please, I welcome my colleagues, Dr. Faiza and Dr. Ahad, and the mic is Dr. Faiza.
Thank you, Dr. Prof. Sarah, for this introduction. And I would like to thank Prof. Asma and her teams for this invitation. Thank you for everyone. Today, me and my colleagues, Dr. Ahad, will talk about the role of machine learning in detecting small and large molecules toxicity in drug development and regulatory affairs. As we know all, the drug development is a complex, time-consuming, and costly process, often taking 10 to 15 years and costing billions of dollars. Drug toxicity refers to the harmful effects a drug can have on the body, which can lead to adverse drug reactions and other health complications. Understanding drug toxicity is crucial for patient safety, public health, and environmental protection, adverse effects can lead to drug withdrawal from market or severe health crisis. As we know all, as a drug development or design, we are scientists. We deal with small molecules and large molecules. The small molecules are the traditional drugs, and the large molecules could be the peptide, antibodies, biologists have distinct toxicity profile and mechanism. While challenging remain, ongoing advancement in data science, computational power, and regulatory framework are paving the way for broader adaptation. The traditional toxicity assessment, as we know all, they rely on the in vitro, the cell culture, and the in vivo animal testing. But there are a lot of challenging in the traditional toxicity assessment. As we said, they are rely on the animal models and in vitro assay, which are expensive, time-consuming, and not always predictive of human outcome. Also, there is a lot. There is the challenging for limited scalability for high throughput screening of compounds. Difficult in predicting rare or long-term toxicities. So the emergency of the machine learning is transforming toxicity prediction and drug development, offering faster, cheaper, and more accurate assessment. So the machine learning offer, as I said, a data-diverse approach to predict toxicity, leveraging large data set and computational power. It can identify patterns and relationships in complex biological and chemical data, enabling more accurate and efficient toxicity predictions. So what is machine learning? It is a subset of artificial intelligence that involves training algorithms to learn patterns for data and making a prediction or decision depend on the data that we are feeded the machine. And there are many types of algorithm machine learning and toxicity prediction. Some of them is supervised learning, regression models, decision trees, or random forest, supported vector machines, and neural network. And the unsupervised learning, like clustering algorithms for identifying pattern in unlabeled data, deep learning like convolutional neural network and recurrent neural network for handling complex high dimensional data. And Dr. Ahad will go through some types and examples and application for the machine learning. For feeding the machine learning, we have a lot of data sources in the toxicity. So if we want to know the chemical structure, we can go to SMILES or molecular fingerprint. There is specific sources for genomic and proteomic data. High throughput screening data, clinical trial data, and adverse event reports. Also, the public database, we can use the PubChem, and Dr. Ahad will go through it and how we can deal with it and what is the information we can have depend on the traditional method and the AI method. Also, there is a lot of application of machine learning and toxicity prediction, but as I said, we deal with small molecules and large molecules. So some of the small molecules, there is a machine learning specific for prediction the hepatotoxicity, which can identify structure feature associated with liver damage. For the cardiotoxicity prediction, there is a model like a HERG inhibitor can predict the cardiac risk. There is a specific machine learning algorithm that can screen for DNA damaging potential to know where is the mutagenicity or and the carcinogenicity. There is a specific machine learning algorithms for the prediction of absorption, distribution, metabolism, excretion, and toxicity. For the large molecule, as we said, could be a peptide or antibodies or that we did in the body, like the immunogenicity prediction. The specific machine learning can predict whether the biologist will trigger an immune response or not, or where is it. Also, the cytokinase release syndrome identify that may cause excessive immune activation. What is it? And the aggregation and the stability that could predict the protein aggregation, which can lead to toxicity. Now, my colleagues, Dr. Ahad, will go through the machine learning and toxicity prediction from regulatory perspective, and will give you informative information and examples for the tradition with dealing with the traditional and the AI tools. So the mic is yours, Dr. Ahad.
Assalamu alaikum. Thank you, Dr. Faiza, for your informative presentation. I will highlight a few examples from applying the machine learning or the AI along with the traditional risk assessment or toxicity assessment from a regulatory perspective. The machine learning is actually a vast changing process, and the toxicities and the genotoxicities are regularly discovered on daily basis. Guidelines and control strategies are also frequently updated. Like if the ICH or EMA or like the pharmacopoeias, they always update the discovery of these toxicities and their controls and limits. In regulatory, we have two definitions for or classifying the genotoxic impurities. We have the genotoxic impurity and the potentially genotoxic impurity. A genotoxic impurity is impurity that been demonstrated to be genotoxic in appropriate toxicity model, such as the well-known bacterial gene mutation test, the AIMS test. And the potentially genotoxic, it means the chemical compound shows a structural error for genotoxicity, but has not been tested in an experimental test model, such as the confirmed genotoxic ones. And the potentially here refers that for being potentially genotoxic, not their presence or not. The possible toxic impurity sources that found in the final API or the final drug substance are the residues of the starting material that can carry out to the final API or to the drug product, the impurities associated with the starting material, the intermediates in the synthesis of the starting material or the active ingredient, also the use of certain solvents or reagents, some degradations, pathway or reaction, side products, and the might be a result of interaction between the starting material or the API with their packaging material. The genotoxic impurity assessment, as Dr. Faiza mentioned, it's actually a combination of both the traditional risk assessment and also the machine learning or the use of AI. In regulatory or in like the quality assessment, it is actually consists of three parts. The first one is the risk evaluation, which means assessing all the risk factors that might lead or produce these potentially genotoxic impurities or the confirmed genotoxic impurities. The second one is the confirmatory testing. Once you have identified your genotoxic impurities, you need to develop and validate a suitable analytical method. The last and important thing is the control strategy, and the control is based on the analytical result you got from the confirmatory testing. In evaluation, we follow the ICHM7 guidelines and other guidelines as well, but this is like the most inclusive ones. It covers the new drug substances, the drug products, clinical development, and the marketing application. Also, the current marketed products. So, like the evaluation for the drugs that want to be intended for the market and the drugs that are already in the market. And what like excluded from these guidelines are like the biological products, peptides, the radiopharmaceuticals, the herbal fermentation products, the crude products that are extracted from plant or animal origins. Also, if the drug substance is itself genotoxic at therapeutic concentration, such for anti-cancer drugs. The control strategies depends widely on or mainly on the classification of these impurities. We have 5 different classes according to the ICHM7 guidelines. The class 1 are the non-mutagenic and carcinogenic too. The control must be based on the compound-specific acceptable limit, the PDE limit, the allowable intake limit. That is based actually on the online trials and the specified value for each impurity. The second type is the class 2. These are the non-mutagens with unknown carcinogenic potentials. These are to be controlled under the TTC limit. The TTC limit is a limit that is based from the duration of a treatment of this medication. The class 3 are the alerting structure unrelated to the structure of the API, and there is no mutagenicity data associated with it. The control could be divided into two. If it's not mutagenic, then it's controlled as a class 5 or as regular impurity. If it is mutagenic, then it's controlled as class 2 based on the TTC limit. The class 4, they have alerting structure, but the same alert in the drug substance, which has been tested in non-mutagenic. It found to be non-mutagenic after the test. So you treat it as a non-mutagenic impurity or regular impurity. The class 5 are the non-structural alerting impurities, sufficient data show that they lack the mutagenicity. These are also to be treated as non-mutagenic impurity and controlled as regular impurity in the final API. Here are some in silico computational tools. Here are comes after you do the risk assessment from your knowledge of the chemical structure, the synthesis, the nature of the API. You also apply along with that the AI or the machine learning implemented tools. It's like a knowledge-based system combined with a statistical system. So you need both to carry out efficient assessment for the toxicities. You have examples such as the Derek Nexus and Sara Nexus, the Readscape, the Multicase, the Tox3, the Lazar, the Case Ultra, the Caesar, the ACD Labs, and the TopCat. The use of in silico tools or software for predicting the toxicity of chemical compounds, it will use a knowledge base, as I mentioned earlier, for the structural alert database. These databases are created by the expert scientists, by data collected online, by chemists also for developing these applications to provide the accurate prediction of various toxicological endpoints. This combine both the applying the structure activity relationship, the SAR knowledge, and also applying the statistical based knowledge that enables AIMS mutagenicity prediction and using the machine learning. You combine both of these knowledge and come to the concluded by the expert opinion. This could be like the head of the scientific office in the company, the drug development team in the company, as and also in this Saudi FDA, it's like the assessor and also the reviewer opinion upon these impurities. I included a few examples just to give you an insight of how to assess the genotoxic impurities and the dossiers that are submitted to the Saudi FDA. We have sitagliptin. In the risk assessment, if you can see in the structure of sitagliptin, there is the chromophore of the imidazole ring, and along the synthesis, the use of certain reagents and solvents, and the pH of the reaction will produce a class 1 nitrosamine impurity that is known as the N-nitroso-triazolopyrazine. As I mentioned, the class 1 are always to be controlled to the PDE limit or the AI limit. And in this case, it is specified to be 37 nanogram per day. And when we did the screening using the in silico software, we found they used the ethane 1,2-diamine, which is a class 2. And the other impurity found was 2-chloromethyl, 5-trifluoromethyl, 1,3,4 oxadiazole. It's a class 3. Both impurities were controlled to the TTC limit. As I said, the TTC limit is a fixed limit from the ICH based on the duration. And we do the calculation based on the drug dose. And the acceptable limit was found to be not more than 15 ppm in the final API. Abiraterone is another example of the drug. You can see from the structure, it does not have a distinctive chromophore or similar to that of sitagliptin or the imidazole. But when we did the screening for like the starting materials, the API synthetic route, we found the very most common impurity, that is methyl paratoluene sulfate. It is a class 2, and again, it is controlled as a TTC, and it found to be not more than 1.5 ppm in the final API. One of the starting materials used was actually a genotoxic one. It's a class 3, and it is also controlled under TTC limit. We have example here for the another starting material is the class 5 non-mutagenic. It's controlled as a non-mutagenic impurity according to the ICH limit, which is maximum not more than 0.15%. Pantoprazole, it's a commonly used medication. Again, you can see the chromophore for the imidazole ring in the middle, also combined with the specific solvents in the synthesis, with reagents. It will produce the NDMA, which is a nitrosamine, a class 1, and its set limit according to the guidelines is not more than 96 nanogram per day. The screened genotoxic impurities were found to be the 4-difluoromethoxybenzene, 1,2-diamine, one of them, and it's a class 3. But you see the difference here. The control limit based on TTC upon the calculation, duration of the use of medication was 6.25 ppm. The other impurity found was, upon screening, found to be inconclusive or outside of domain. These impurities are to be treated as regular impurities and not more than 0.15%. Tigecycline, it is the most common used antibiotics for treating certain diseases. You can find like it produces different types of nitrosamines, and they are the class 1s. The control of them is fixed according to the AI limit, each specific for each impurity. And here again, after the risk assessment of the tigecycline and the nitrosamine they produce, when we did the screening, we found 2 impurities, the 9-nitrominocycline disulfate and 9-aminominocycline disulfate. Both were class 3. Upon the calculation, the TTC limit was to be not more than 15 ppm in the final API. I would like to sum up the combining of the traditional risk assessment of toxicities with the AI. They will give you efficient information or give you overall pictures of what you could expect from this drug substance. It will help you to develop your own database, help you in the assessment. You can have like a very good chemical background and knowledge of different APIs, the different use of solvents, agents. You can expect certain outcomes, degradations, impurities, side products, and so on. The use of both techniques for assessing toxicities is crucial, especially for the drug development and also for the assessment of the dossiers submitted to the Saudi FDA. Thank you.